My name is Alice Prohaska. I'm the principal of Somerville College, and we are enormously proud of running this annual lecture in memory of Dorothy Hodgkin, who was, as all of you I'm sure will know, the first and so far only British woman to win a Nobel Prize for Science. She was a student at Somerville, a fellow of Somerville, a professor in the University of Oxford, one of our most distinguished ever alumni, and the college is enormously proud of her and very, very honored to be able to honor her memory with this annual lecture. We are very, very pleased today to welcome our speaker, who, following in Dorothy Hodgkin's footsteps, is also a Nobel Prize winner, Dr. Venki Ramakrishnan. And I think I'm not really supposed to say that he's actually Sir Venki, but he doesn't like to use the title. Knighted in the New Year's honours, just the latest of many honours that he has gathered for his science. Venki started his career in India, has worked a large part of his career in the United States, and now is working in Cambridge at the MRC Laboratory of Molecular Biology. He, amongst his many honors and distinctions, he won the Louis Jeantet Prize for Medicine in 2007, the Nobel Prize for Chemistry in 2009, and then the Padma Vibhushan Prize in India, which is a prize of immense, to honor Indian citizens or people of Indian origin of immense distinction. The honors keep on coming inexorably, and despite your great modesty, Venki. He's going to speak to us now about ribosomes, and I'm going to leave you without more ado to take the floor. Thank you very, very much.
Thank you very much. It's a great pleasure to be here, and it's a particular honor because Dorothy Hodgkin was one of the pioneers of our field and really made breakthroughs in tremendously difficult circumstances, you know, that we just cannot help feeling awed by, given all of the resources and technology that we have at our disposal today. So I'm particularly honored and grateful to have been chosen to give this talk. Now, you may have wondered what these people have in common. The people on top were literary giants, and these are famous composers. And Ramanujan was a self-taught mathematical genius who was the first Indian to be elected Fellow of the Royal Society, and also the first Indian to be elected a Fellow of any Oxbridge College. He was a Fellow of Trinity College. Now, what you can see from the birth and death dates that I've conveniently put up there is that they all died tragically young. And you wonder what they might have accomplished if they had lived to what we consider a normal age. And today, and they all died of infectious disease. And today, if we have an infection, we expect to go to our GP and get antibiotics and get treated. And the question is, are those days really over? Well, even today, there are 100 million people who have active cases of tuberculosis, which cause 2 million deaths a year. Now, you might think that tuberculosis is largely restricted to the third world, but actually, this is an article from the Times showing an outbreak of TB in Leicester. And I know parts of Leicester resemble the third world, which are the parts I like because they have excellent restaurants. But the fact is, you know, Leicester is in Britain, And so it is rising in the West, and it's partly coupled with the rise of AIDS, which makes people susceptible to TB. Now, you might still say, well, all right, but TB has cures, and that's largely true, although now, just last December, when I was in India, there was a headline which described TDR-TB, which is totally drug-resistant TB, which is absolutely untreatable by any known antibiotics. So these people are now back in the 19th century, as it were. They have to rely on their immune system to cope with it and either get cured or die. But leaving aside TB, there's another organism which is of more immediate threat to all of us, and that's Staph aureus. Aureus means gold, and it has to do with this pigmentation, this gold pigment. And Staph aureus is a bacterium that likes to invade wounds. And so it's very common to have Staph aureus infections after surgery. And when penicillin was first made available, it was found that nearly all of Staph aureus could be treated with penicillin. That is, there was no resistance. And what the black squares show is the rise of resistance in hospitals, which is where surgery takes place. And what the white squares show is the rise of resistance in the general community. So there's a lag of a couple of decades, but today, nearly all Staph aureus is resistant to penicillin. However, Staph aureus can be treated by other drugs, but there are now strains of Staph aureus that can only be treated with one antibiotic, vancomycin. And these are called methicillin-resistant Staph aureus, or MRSA. And MRSA can be quite deadly. And just now, 25,000 people a year die in Europe alone. So clearly, we still are facing a problem with infectious disease. It's an ongoing battle. So the question is, how did the notion of fighting infections with medicines occur? Well, in the modern era, it first occurred to Paul Ehrlich, who coined the phrase magic bullet. And the reason for that was that he noticed that when you use certain stains, these rod-like bacteria would take up the stain much more than these human cells. And so his idea was that if you had something that was toxic to the bacteria, and possibly it might be toxic to humans as well, but let's say it was preferentially taken up by bacteria, then you would have a compound that could kill the bacterium without killing the host cell, without killing the human cell. And that was the idea that occurred to Ehrlich. Now, he himself had very limited success with this idea. He found a rather toxic mercury compound that was used to treat syphilis. It was an arsenic compound. Sorry, not mercury, but arsenic. And but his protege, Gerhard Domark, worked for the Bayer Chemical Company. So he had access to thousands of dyes and other organic compounds. And he did a systematic search and came out with a red compound called Prontosil, which was effective against Streptococcus and other bacteria. And it was the first effective drug against fatal infections. Prior to that, if you had a cut, let's say you were a child in a playground and you cut yourself, and you got a Streptococcus infection, the infection would simply spread, and there was no recourse to it. And often people would have to be amputated. And interestingly, he saved his own daughter from having her finger amputated by the use of Prontosil. But he had a couple of unfortunate things. One was that the red compound Prontosil was actually broken down in the body into a colorless compound called sulfanilamide, which was already known. And so his patent on Prontosil was voided almost a year after he had discovered it. So he got no money out of Prontosil. The other problem he had was he was awarded the Nobel Prize in 1938, which was during the Nazi era. Now, Hitler didn't know the difference between Norwegians and Swedes. And when the Norwegians awarded a peace prize to a German pacifist who didn't like the Nazis, he was outraged and he forbade German citizens from accepting the Nobel Prize. So as a result, Gerhard de Marck could not accept it. But after the war, the Nobel Foundation very generously invited him to the next Nobel ceremony and presented him with the Nobel certificate and medal, but told him that, alas, his prize money had been returned to the general funds, and so he couldn't have it. So that was Gerhard de Marck, but he did start off a revolution, and sulfur drugs, I mean, many of us have, certainly some of us in this audience are old enough to remember having taken them. And actually, the successors of sulfur drugs may have saved my wife last year when she came down with a fungal pneumonia. And so they're still very widely used to save lives. Now, sulfur drugs are synthetic chemicals, but there was another approach that became very powerful in the treatment of infections, and that was the discovery of naturally made antibiotics. And that came about as a result of the discovery of penicillin by Alexander Fleming, who noticed that when he left a plate outside, it got contaminated by a mold spore, and he noticed that bacterial colonies could grow far away from the spore, but they seemed to be inhibited by something that the spore was secreting, and he correctly surmised that it was secreting a compound called penicillin, and went on to suggest that it might be useful. But he never actually followed it up himself. And the story of penicillin and its utility for human disease is actually a triumph of Oxford. And that started off when Florian Chain figured out how to actually purify large quantities of penicillin. And this required a tremendous effort, including someone named Heatley, who worked with Florian Chain. And at the time penicillin was first made, it was so precious that they extracted it from the urine of the first patient that they gave it to, and had to repurify it from the urine and then sort of give it back to him, because most of the penicillin would be excreted. And so, but because it was so precious, as soon as he started improving, they stopped the treatment, and he got a relapse and died. And there's a famous medical saying, which is that the treatment was a success, but the patient died, which is such sad irony. But penicillin, of course, went on to save many lives, but it was important to know its structure, because penicillin is actually only effective for gram-positive bacteria. It's not effective against gram-negative bacteria. And in fact, semi-synthetic forms of penicillin, like ampicillin or various other compounds, are the ones that are used today. In fact, pure penicillin is very rarely used for treatment. And in order to know that, you needed to know its structure. And that, of course, was one of the major discoveries of Dorothy Hodgkin. And here is the original electron density map of penicillin. And when she elucidated the atomic structure, this was done when chemists were still not quite sure what the structure was. So it was actually a crystallographic triumph. Very often, crystallography would determine the three-dimensional arrangement of a chemical structure that was already known, in the sense that the bonds between the atoms were already known. But this unusual four-membered ring, the beta-lactam ring, was first actually discovered by crystallography. So it's a great triumph. And of course, all of us are very pleased that the post office has decided to recognize famous British scientists, and among them Dorothy Hodgkin. Now, penicillin and its derivatives are useful for a variety of infections, but they're not useful against TB. And Selman Waxman was a microbiologist working in New Jersey, in Rutgers, and he heard Fleming give a talk, and he said, you know, if these Englishmen can do something like that with a mold, we ought to be able to do a lot better with this variety of soil bacteria. So he had a certain attitude, let's say. And he convinced his graduate student to survey a wide variety of soil bacteria. And in fact, they all come from one genus, which is called Streptomyces, and they're different species of the same genus. And they found that an organism called Streptomyces griseus actually was not only effective against a wide variety of other bacteria, but it turned out to be the first effective antibiotic against tuberculosis. As you can see, as you increase the concentration of streptomycin, the bacteria are unable to grow. There's a story about Schatz as well. Schatz actually did most of this work himself in a room in a different building with very little contact from Waxman. And Waxman was awarded the Nobel Prize, and after that he also had the patent with Schatz, but somehow there was a great acrimony between them, and Schatz went on to sue Waxman for credit over the discovery. So it's a lesson that you really should give credit to people who actually do the work, or one day you may find yourself in court. So anyway, streptomycin was the forerunner of a lot of antibiotics, because once the idea that the soil bacteria could be sources of useful compounds became known, companies became very interested. And a huge number of useful antibiotics were discovered as a result of that. And the thing that these antibiotics all have in common is they all work by blocking protein production in bacteria. So I've sort of told you a lot about antibiotics and their discovery, but now I'm going to switch gears and talk about how proteins are made. Because if it's blocking protein production in bacteria, you have to understand how bacteria make proteins. And it turns out that process is actually universal. All of us make proteins in essentially the same way, including bacteria. Now, we all know that genetic information resides in DNA, but largely what genes contain is information needed to make proteins. This is not entirely true. We're finding more and more examples of genes that don't code for proteins but do something else. But let's take it for granted that most of them, the important role that they have is to make proteins at the appropriate time in the cell. And the way that's done is that DNA is converted into RNA, and RNA is simply a copy of DNA. And by that I mean it has the same building blocks as DNA, which are the 4 bases that go to make DNA. And RNA consists of the same types of bases. But proteins are chains of amino acids, and there are 20 types of amino acids. So you have to go from a 4-letter alphabet to a 20-letter alphabet to get a protein. And if you ask what are proteins, well, proteins do a myriad array of functions in the cell. For instance, the only reason you're able to look at me or the screen is there's a protein called rhodopsin in your eyes, which detects light and then sends a signal to the brain. Proteins in your red blood cells carry oxygen. The proteins that fight off infections, antibodies that fight off infections are proteins. So, and the skin and hair that give you form without which you'd just be a blob are also proteins. And so proteins carry out thousands of functions. Now, the way that you get from a DNA, from the gene to a protein, is through an intermediary molecule which has 3 bases that recognize 3 bases here, just as the bases in the double strand of the helix recognize each other. That was the famous discovery of Watson and Crick, that A pairs with T and C pairs with G. So this molecule, tRNA, recognizes a triplet and brings the appropriate amino acid at the other end. And this amino acid somehow has to be joined to the next amino acid, which codes for the next 3 bases, and so on. I'll come to that later. But clearly, this sort of thing doesn't happen by itself. And cell biologists discovered that newly made proteins were localized on these blobs on something called the endoplasmic reticulum in the cell. And these blobs were isolated, and they were all particles of about 250 angstroms in diameter. And they all consisted of 2 halves, a large and a small subunit. And because they consisted of 2/3 RNA and 1/3 protein, they were called ribonucleoprotein particles of the microsomal fraction. But if you wanted to say that every time you wanted to refer to it, it would be rather tedious. And so in the late 50s, Howard Dinsis gave them the name ribosome, and that's the name that stuck ever since. If you ask what are ribosomes made of, they're made of about 50 different proteins and 3 large pieces of RNA that go to make up the 2 subunits, the large 50S subunit and the small 30S subunit. And this is what the ribosome looks like in an electron micrograph from the sort of 90s. We have much more detailed pictures now. So if you ask now, how does a ribosome synthesize proteins? You can see what happens is that the small subunit binds the genetic message, and this is that tRNA molecule that I showed you, but simplified. And it's brought along the first amino acid, and then the next tRNA comes in, recognizes the next bit of genetic code, and then the ribosome catalyzes a bond formation between the first and the second amino acid, and then the whole assembly has to move so that, and which leads to the ejection of the first amino acid, but then opens up a slot for the third tRNA, which comes in and brings in the third amino acid, and so on. So you can see that as you're reading the message on the mRNA, you're then stitching together the correct order of amino acids to make the protein. Okay? Now, I've shown you a very simplified cartoon, but a more realistic picture comes from cryo-electron microscopy. And this is what it looks like. The mRNA snakes around the head of the small subunit, and here are where the codons, which are the triplets of bases, are presented to the tRNA. This is the growing protein chain, and this is the new amino acid that's about to be attached to the growing protein chain. Now, if you want more detail than this, you need to get an atomic structure. And this is you cannot use a microscope to get this sort of structure, because what a microscope does is the following. This is what we would normally do for objects that are small but not really small. What you do is take an object and you would use a lens and get a magnified image. Now the question is, how does a lens work? What a lens does is it gathers scattered rays from the object and it recombines them to make an image of the object. Now, all of you are doing that at this very minute because you have a lens in your eye, and the scattered rays from the screen are going into your eye, and the rays are scattered all over the room, so you're really only sampling a small fraction of the scattered rays. So you don't have to sample all of them, but you have to sample enough of them. And the lens in your eye then recombines those rays to form an image in your retina. Now, the image in your retina is actually much smaller than the image on the screen. But if you were to reverse the positions of the lens of the object and the image, then you would get a highly magnified image of a very small object. So that's how magnifying glasses work, for instance. Now, and that is how a microscope works. But there's a law in physics which says that you cannot magnify, you cannot discern details that are smaller than the wavelength of the radiation or light that you're using. And light has a wavelength of about 5,000 angstroms. And the distance between atoms is about 2 angstroms. And so clearly, you're not even in the ballpark. But there is a radiation that is in the right ballpark, and that's X-rays. And that's the technique pioneered by Bragg and subsequently by Dorothy Hodgkin and Max Perutz and others. And that's crystallography. So you could form crystals of your object. And what you would then do is take your crystal and hit it with a beam of X-rays. And then you would detect those scattered X-rays. And then what you would do is you would do what a lens does, except you would do it in a computer. You would take those scattered X-rays and recombine them to make an image of the object. I'm glossing over some of the details, such as knowing which part of the light wave you're detecting for each of these scattered rays. And that's a complicated problem. But fundamentally, you're doing in a computer what a lens does in real space. And for a crystal, the scattered rays are concentrated in these spots. This was actually a discovery of Lawrence Bragg. And all of crystallography depends on the measurement of these spots and then combining them to make an object. So you need two things for this. One of them is, of course, you need crystals. And crystals of small molecules are much easier to obtain. But the ribosome is a large and somewhat floppy object which has 500,000 atoms in it. And it's about 2.5 million Daltons in atomic weight. So it is a giant molecule. And so it was a major breakthrough when Ada Jonat and Heinz Günther Wittmann reported true three-dimensional crystals of the large subunit in 1980. And then about 5 or 6 years later, a group in Russia headed by Marina Garber and Murat Yusupov and Trakhanov produced crystals of the small subunit as well as the entire ribosome from a different species, Thermus thermophilus. Now, these crystals didn't diffract very well. That is to say, if you were able to obtain an image of the object from those crystals, it would be rather fuzzy. It would be too fuzzy to actually determine a molecular structure that would tell you where the atoms were. So two other advances had to be made. And one is the technique of cryocrystallography, which, Elspeth Garman, I believe, was a lecturer in the series. And yes, and she has done a lot of work on cryocrystallography here at Oxford. And this is a technique that Ada Jonat collaborated with Hakan Hope on to apply it to ribosomes, to allow ribosomes to take very intense X-ray sources. And so that was needed, and that allowed you to actually collect data from ribosomes. And then by looking at lots of different species, she found a species of 50s subunits that actually diffracted to 3 angstroms resolution. And the rest is a somewhat complicated story that's worthy of a book, I think, because the 50s subunits that were actually discovered by Ada Jonat were actually solved by Tom Stites and Peter Moore at Yale. It's a bit like penicillin. Fleming discovered penicillin, but really to actually produce penicillin and make it work and solve its structure took other people really to do it, but it doesn't really take away from Fleming's credit. So, and you know, we have the same situation with Rosalind Franklin and DNA. So these things happen in science. So anyway, when I entered the field, there were no good crystals of either the 30S subunit or the whole ribosome, and that's where I actually focused my efforts. The other thing that you need are very powerful sources of X-rays, which are called synchrotrons. And much to my chagrin, the British synchrotron was built near Oxford rather than in Hinxton, where I would have preferred it. But nevertheless, it's easier to drive 2 or 3 hours than to take a flight to Grenoble. But these are the synchrotrons that we've largely used for our work, although we've started using the diamond synchrotron near Oxford more recently. So this is what the so I joke that just like the US Navy advertises that you can join the US Navy and see the world, I say you could join a ribosome lab and see the world, because you could go to all these synchrotrons and all these exotic locations. But actually the Navy, of course, is lying to these poor, naive 18-year-olds who merely see the inside of battleships. And what my people actually see are the world's synchrotrons. And what they really see is this. And this is a synchrotron hutch at the Swiss light source. And the X-rays come out from here. And this is something that keeps the crystals very cold by having a cold gas of gaseous nitrogen. And the crystal is here. And this is a CCD detector that detects X-rays. Now, if your experiment works, what you end up is with something that looks like this, which is a three-dimensional image of the object. It doesn't tell you what the molecular structure of the object is, unless you have very, very high resolution and you can actually see the atoms. But very few structures, certainly in the kinds of things we work on, reach that level. So how do you go about solving this structure? Well, this is a three-dimensional object, and it's a little bit like solving a very large jigsaw puzzle in 3 dimensions. Now, those of you who like to solve jigsaw puzzles know that when you want to solve a puzzle, you start off with the pieces that you know. You recognize the edges and the corners, and you work with those first, and then you start building areas that are easily recognizable, and then save the really hard bits for the very end. And that's exactly the way these structures are solved. What you do is you look around and see if there are areas that you can recognize. And if you zoom in on this area, you suddenly see that there's a strand here which has these very regular bumps. And there's another strand across from it that also has similar bumps. And if you know your structure, what you recognize is that this is a piece of double helical RNA. It's a piece of double-stranded RNA, and you have sugar and phosphate backbones alternating. And so suddenly you realize that you have converted a little piece of that three-dimensional image into a molecular structure where you've placed all of the atoms. Now, this is only a small part of the overall structure, so you then have to keep on building as much as you can, and this can sometimes take months, depending on the structure, or it can take actually even minutes for relatively easy structures to high resolution, where you start a program and go off and have a cup of coffee. That's how things have advanced these days. But the ribosome was done the hard way by about 5 people working together for many months in order to get the structure. So in 2000, there were structures of both the large and small subunit. And then a few years later, we obtained a structure of the entire ribosome with tRNAs and the genetic, a little piece of that genetic message bound. So we now have atomic structures, and we're in a position to understand how antibiotics actually block the ribosome from functioning. So I forgot to show you a movie, so I'm going to just go back. So this will give you a rough idea of how ribosomes work, and that's important because antibiotics block almost every step of the process. So what happens first is that the small subunit binds the genetic message with the help of 3 proteins and brings the first tRNA with that first amino acid in it. And then the large subunit joins. And at some point, this factor called IF2 leaves the ribosome, and the next tRNA comes in, and it's brought by another protein factor, which actually hydrolyzes a molecule called GTP and releases the tRNA so that it can then add its new amino acid to the first one. That's that linkage between the first and second amino acid. And then the tRNAs move first with respect to the large subunit, and then with the help of this factor with respect to the small subunit. So remember this movement, because I'm going to show you how antibiotics can stop that movement a little later. And then the next tRNA comes in, and then the whole process keeps on going. Now this process happens at the rate of 20 amino acids per second, and as the protein chain forms, it emerges through a tunnel in the large subunit and folds into its 3-dimensional structure, often spontaneously, but sometimes it takes other proteins to help it to fold. And then a protein recognizes a special triplet which says this is the end of the message, so cleave off the protein from the ribosome. That's called a release factor, and the newly made protein then emerges from the ribosome, and then a recycling factor then binds to the ribosome, splits it apart so that the entire process can then start all over again. So that is the process, and antibiotics stop virtually every step of that process. Now, what are these processes? Well, I showed you several stages. But one of the first stages is recognizing the code. And what this involves is where the ribosome is waiting for a new tRNA to come in to bring the next amino acid to add to the growing polypeptide chain. And it has to choose from a pool of tRNAs, which all have a variety of amino acids. But it has to choose just the right one that corresponds to that code on the genetic message, so that its anticodon and this codon will base pair correctly. And the way that's done is that, well, I just showed you this movie, so I'm going to skip. The way that's done is the EF2 tRNA actually comes in, and if it's the right codon, then the tRNA bends and is stabilized by bases in the ribosome right here. And that leads to a series of changes which result in hydrolyzing this molecule of GTP into GDP, which results in this protein changing its shape and leaving the ribosome, allowing the tRNA to move in and then add to the growing polypeptide chain. And here is a movie that describes that same process in a little more detail. So you have the tRNA that comes in, and it binds. And when it binds, the tRNA samples a variety of conformations. It can bend and sample the codon. And if it happens to be the correct codon, then you have a change in these ribosomal bases which recognize the pairing between the codon and the anticodon and stabilize that particular tRNA and allow the tRNA then to contribute its amino acid to the growing polypeptide chain. So how do antibiotics work? Well, if you have a structure, you can solve the crystal structure in complex with the antibiotic. And what you then can do is take a difference between the structure with the antibiotic and the structure without, and it will give you an image of the antibiotic. And you can then interpret that in the molecular structure of the antibiotic. And so here is an antibiotic called spectinomycin, which I'll return to in a minute, which actually binds in a little hinge point in the ribosome. So this is the whole structure of 1 million Daltons, and this is only a few hundred Daltons, and yet you can, using crystallography, determine exactly how that antibiotic interacts with the ribosome. So doing this, we were able to determine the structure of lots of different antibiotics bound to the 30S subunit, and the Yale group and others determined structures of antibiotics bound to the large subunit. So here is two very useful antibiotics. Tetracycline, which is still a very widely used broad-spectrum antibiotic, and doxycycline, which many of you may have taken if you visit tropical countries because it's a malarial prophylactic. But interestingly, there's some ideas for using it to treat elephantiasis. You know, you may have seen the movie Elephant Man, which is, caused by filaria, which is actually a parasite. But it turns out that there's a bacterium inside the parasite that is a symbiont. So the parasite actually needs the bacterium, and the doxycycline will kill off the bacterium. So if you can give it at an early stage before all this tissue damage has occurred, you could actually maybe prevent filariasis in the future. Now, how does tetracycline work? I've shown you how the tRNA is delivered to the ribosome, and it turns out that tetracycline binds right here, right between the codon and the anticodon, so that it would actually prevent the tRNA from coming in and binding and then delivering the amino acid to the next growing polypeptide chain. So it would block protein, it would stop protein synthesis in its tracks. The other antibiotic that we worked on were aminoglycosides, of which there are several. These are not compounds. Gentamicin is often used in eye drops, but these are not compounds you would normally want to take. If you're having to take these, it means you're in very bad shape, and other antibiotics have failed because they're rather toxic. And if you see how they work, the way they work is they bind in that region where tRNA and mRNA interact. So the tRNA recognizes the genetic code, and here is where paramomycin, one of the antibiotics, works. And what it does is it makes that recognition a little easier, significantly easier, so that the ribosome starts recognizing incorrect tRNAs, and so makes proteins that are essentially defective because they often have the wrong amino acid. And the other antibiotic is streptomycin, which I mentioned was one of the first of this class, which was discovered by Waxman, the first effective drug against TB. And the streptomycin binds here, and the way it works is that when tRNA binds, there's actually a movement of the 30S subunit to close in around the tRNA. And streptomycin favors that closed form, and so also allows incorrect tRNAs to be recognized and increases the error rate of protein synthesis. Finally, I'll come back to spectinomycin, which is the antibiotic that inhibits movement. This is the one that I showed you in that first image. Now, translocation occurs by movement of the tRNAs first in the large subunit and then in the small subunit. Now, this intermediate state where they've moved with respect to the large subunit but not with respect to the small subunit also involves a rotation of the two subunits relative to each other, as well as movements within the small subunit. And only last year, we were able to get a crystal structure, not of the translocation state, but a state further down the pathway, but the structure is thought to be very similar to that snapshot of that intermediate state, because we have a tRNA that's moved with respect to the 50S subunit, but not with respect to the small subunit. And the way that works is when this factor binds, you see this movement. But notice that there was also movement of the small subunit relative to the large, and this is what's called a ratcheting movement. It was first discovered by electron microscopists and Joachim Frank and his colleagues. And so you have this ratcheting movement. But the small subunit doesn't just ratchet. The head of the small subunit also swivels. And so the way that happens during translocation is that you have the head of the small subunit swiveling with respect to the rest of the molecule. And in red is where spectinomycin binds. And if we analyze this swiveling movement, we see that spectinomycin would actually inhibit the swiveling movement. You would get a clash of atoms, and so the swiveling would not be able to take place. And so you see that movement can be inhibited by these antibiotics, and by inhibiting movement, you're essentially blocking protein synthesis because that movement is required for ribosomes to go from one position to the next. Now, I want to talk about a class of very important antibiotics that were not done in my lab, but at Yale. And these are antibiotics that bind to the large subunit, which bind at the site where the peptide bond is formed. This is where amino acids are added to the growing protein chain, and also at the site where the protein chain has to go through as it emerges from the tunnel. So this is the peptidyltransferase center. And just to show you what it looks like, here is the ribosome in what's called the molecular biology textbook view, with the 50S subunit on top, the small subunit at the bottom, and the tRNAs in between. And I'm going to zoom into the peptidyltransferase center. And here is where the growing polypeptide chain would be, and this is where the new amino acid would be added. So this is the first amino acid, let's say, and this is the second amino acid. And now a bond has to be formed between these two, and the protein chain has to emerge through this little tunnel here. And what you can see is that chloramphenicol binds where the A site tRNA, the new amino acid, would be. So it would prevent the binding of the new amino acid. And so you would prevent peptide bond formation from occurring. Even if the tRNA were delivered, you couldn't get the new amino acid in the right position to form a bond because the antibiotic would be sitting there and preventing it from binding. Then other classes of antibiotics called macrolides bind very nearby. So here is chloramphenicol, which still is used for typhoid. It's not a very good antibiotic. It's nasty because it gives you anemia. But people like me who grew up in India have had to take it if we had typhoid, and I actually took it when I was a child. And so it may have saved my life, actually, this little molecule here. And these are other antibiotics which are still widely used, which are called macrolides, of which erythromycin is the most common class. And you can see that they bind at the entrance to the tunnel. So even if a protein chain bond can be formed, the protein chain is unable to emerge through the tunnel, so the ribosome sort of gets gummed up, if you like. And this is all of these antibiotics have this ring structure called the macrolide ring. So in the last few minutes, I want to say how these structures are being used in new companies to actually make important new compounds that could be useful. So Tom Stites, who did the large subunit and many of these antibiotic structures at Yale, founded a company in New Haven called Ribex Pharmaceuticals. And what they decided was something rather clever. They noticed that here is where chloramphenicol binds, and this is where erythromycin binds. They noticed that these two are very different binding sites, but they're right next to each other. Now, it turns out that erythromycin does not work very well against many strains of pathogenic bacteria. These are clinically relevant bacteria which are resistant to macrolides. Okay? So you can see in red it shows that the minimum inhibitory concentration required for the bacterium is quite large. But what they decided to do was they said, let us see if we can make compounds that span both sites. And so they made a compound. I have no idea what its structure is because it's proprietary, but that's a sort of general outline. Okay? And then they actually soaked that compound into ribosomes and determined its structure, and this is how it binds. It binds exactly as they had predicted it would bind because it was designed using erythromycin and chloramphenicol. And when they tested it against all of those resistant strains of bacteria, you can see that now this compound is effective against strains that were resistant to normal macrolides. And they're in the process of trying these in clinical trials, and in fact, they now have compounds, not this particular one, but they have other compounds which are in phase 3 clinical trials. So that is to say, they've already been shown to be not toxic, or at least not immediately toxic in humans, and also efficacious. And now they're in the process of doing phase 3 clinical trials. And as you can see, there are now other classes of antibiotics which bind all around that area. So you could link up different classes of antibiotics and make varieties of new compounds. So the hope is this will be an interesting new strategy to target the ribosome to come up with clinically important drugs. Now, ribosome antibiotics work because of small differences between human and bacterial ribosomes. So they're able to bind tightly to bacterial ribosomes, but not so tightly to human ribosomes. So if you give them at a very low concentration, they'll mostly bind to the bacterial ribosome and kill the bacteria rather than kill us. But many antibiotics are not so selective, and that's why they're significantly toxic, like chloramphenicol, for instance. So no matter how many antibiotics we have, it's unfortunately a fundamental law of natural selection that resistance will emerge. And resistance can emerge in a variety of ways. I've shown you many examples that antibiotics work by a tight fit between the antibiotic and its target pocket in the ribosome. And this is how all antibiotics work, whether they target the ribosome or not. They work by targeting a pocket that stops the functioning of some important molecule in the cell. And you can disrupt this precise binding between the antibiotic and the pocket in several ways. One is you could break down the antibiotic, and that is how penicillin resistance works, by enzymes that degrade penicillin so it no longer has the right shape to bind tightly in its pocket. Many other resistance mechanisms work by modifying the antibiotic, by adding extra little groups so that what fit tightly like a hand in a glove now no longer fits because you have extra little bumps that don't fit in the pocket because you've modified the antibiotic. And finally, another important mechanism is that many bacteria have pumps that pump out compounds it doesn't like, including antibiotics, and these are called efflux pumps, and they're an important class. But the problem is, no matter what the mechanism is, you will eventually get resistance. So this is an ongoing battle. And it takes over $1 billion and over a decade of research to even come up with one new antibiotic. And so this is not a problem that science and drug companies can solve alone, but it's going to require a concerted attack by society. One is you need surveillance so that infectious epidemics are quickly seen and stopped. You need infection control, which is proper sanitation, prevention of unhygienic techniques. If you go to any hospital in Britain now, you'll see a big sign which says, wash your hands, okay? That's prevented more infections than, antibiotics would have cured. In fact, the rate of MRSA infections dropped dramatically in British hospitals when that policy was applied. And finally, you need to promote rational use of antibiotics. In America especially, but less so here because of the NHS. People go to their doctors with a flu or a runny nose and demand antibiotics. And in fact, all this does is spread resistance because these antibiotics don't do anything to cure you. And they also use antibiotics, and this is, I'm afraid, true all over the West, is they're used in animal feeds to fatten farm animals. And that's a terrible use of antibiotics. And I'm surprised it's still legal. So, but on the other hand, these are social things, but science can do our bit. We can understand how bacteria actually cause disease. We have millions of bacteria in us, and probably we wouldn't be alive without them. So most of these bacteria are beneficial. So it's only certain bacteria that cause disease, and we need to understand why they cause disease. We need better diagnostics and vaccines. And this is the part where structural chemists like myself could actually be of some use. So thank you very much.